Hello, everybody. Welcome back again. We are continuing with our progress in the discussion of cells and cellular structure. And this time we are going to focus very specifically on the membrane. You'll recall in the previous presentation, we talked about all the organelles that are present in eukaryotic cells particularly, and many of those have membranes associated with them. Plus, we also need to remember that every cell has a plasma membrane. So it appears that membranes must have some very important function associated with cellular processes, and that is in fact true. Nothing surprising there. This is, again, something that we need to recognize. If there is something present in a cell or something present in an organism, it's there for a reason. It has function. Otherwise, natural selection and evolution would have ultimately removed it, because there is a basic principle involved. You could think of it as a cost-benefit analysis if you are so inclined, because that's in essence what it is. And it basically goes like this: if it costs more to maintain something than the benefit you get from it, natural selection and evolution eliminate it. So the fact that we have all of these membranes, that all cells have membranes to demarcate the inside of the cell from the outside of the cell, and that many of the compartment or the organelles in eukaryotic cells are defined by the presence of membranes, suggests that those membranes have very, very important functions. And that function must in some way be associated with its structure. So we have a twofold goal in this presentation, in this chapter, chapter 7, to identify the structural feature of the membrane that gives rise to all of the functions, and then to focus in very specifically on a couple of functions, most notably transport, that turn out to be critical to understanding the chemical behavior of a cell. Because after all, if a cell is going to carry out chemistry, it needs to have available the reactants for the chemical reactions that it's going to carry out. And many of those may not be the product of the cell itself. And also it needs to be able to export some of the products of those chemical reactions. So all of that is wrapped up in an understanding of the basic structure of a membrane and what that tells us about the function, the potential function of the membrane. And then we're going to focus in very specifically on membrane function with a very sharp focus on transport. So that being said, let's get started. So the first thing to remember is that every cell has a plasma membrane. It is the line of demarcation that separates the inside of the cell from the outside of the cell. And one of the things that we need to bear in mind is that if it was a hard barrier, meaning nothing could get across the membrane, then whatever was inside the cell would remain inside the cell. Whatever is outside the cell would remain outside of the cell. There would be no communication backwards and forwards, and very quickly the cell would probably become non-functional. It would die, because things do need to get into cells and things do need to get out of cells. And given that most cells are aerobic, that is, they require oxygen to carry out their basic functions in terms of generating energy, then it stands to reason that oxygen needs to cross and carbon dioxide needs to get out, because that would be the product of cell respiration. But we also know that many, many other molecules move into and out of cells. But not everything has the capacity to move in and out of cells randomly. And that's because cells have a particular property. Now, when we talk, or more specifically, their membranes have particular properties. Now, when we talk about things moving across membranes, the term that we use is permeable. Permeable means that the molecules can move through it. And so we talk about the idea of permeability. Is the membrane permeable? And what we notice is that cell membranes and membranes in general, including the membranes that surround organelles inside eukaryotic cells, are not infinitely permeable. The term that we use is selectively permeable, meaning that some things can move freely backwards and forwards, other things not so much. And because of that, it allows cell membranes to become regulators of what moves into and out of cells and what moves into and out of organelles inside cells. And we've already seen that, because when we talked about the nucleus last week, we talked about the presence of nuclear pores in the nuclear envelope. And I pointed out that those are gatekeepers. They allow some things in and they allow some things out. And they are the only way of things moving into and out of the nucleus. So the presence of those nuclear pores in the nuclear envelope, which remember is a double membrane structure, regulates what moves into and out of the nucleus. And all membranes serve the same function in cells, whether they be the plasma membrane or whether they be the organelle, the membranes of the organelles that represent these compartments inside a eukaryotic cell. What we also know is that membranes serve as a very important anchoring point, not only for proteins that may be involved in moving things in and out of the cell, but also for many of the proteins that are associated with cellular activity. So we will discover when we do chapters 9 and 10 and talk about cell respiration and photosynthesis, that a large number of the enzymes that are associated with the chemical reactions of those processes are themselves associated with cell membranes. They are not freely floating around inside the organelles. They are actually associated with the membranes, either attached to the surface of the membrane or in some cases inserted into the body of the membrane. They are an integral part of the membrane. This is an idea that we will focus a little bit more on as we move through the presentation this morning. So we have to recognize that a membrane is more than just the phospholipid bilayer that we identified back in chapter 5 when we were talking about lipids. The fact that phospholipids are the base component of every membrane in the form of these phospholipids that form the bilayer, the phospholipid bilayer, that really does represent an outstanding separator between two bodies of water, because of the fact that the inner part of that bilayer is extremely hydrophobic because of the hydrophobic nature of the fatty acids of the phospholipid. But it also means that they can also interact with water because of the hydrophilic, we say heads, but the hydrophilic portion of the phospholipid. Remember, a phospholipid is amphipathic. It has both hydrophobic and hydrophilic regions. And this is what results in the spontaneous formation of the bilayer. And it was the recognition of the fact that membranes actually contain phospholipids very early in the investigation of cell composition that led to the discovery of the presence of proteins. Not because they were easy to identify, but in many cases because the initial ideas of where those proteins might be, once it was recognized that proteins were part of membranes, proved to be inaccurate and forced people to really start thinking about the nature of the proteins and where they might be with regard to the phospholipids. So that's something that we're going to discuss in a little bit more detail over the course of the next few slides. So let's move on. So this is a very complicated diagram that illustrates the nature of a phospholipid bilayer with proteins inserted into the bilayer. So the phospholipids themselves are represented by the green strands here. These are the fatty acids of the phospholipids. And the hydrophilic heads are these, I'm going to say gold or yellow balls. That represents the hydrophilic heads. And you can see that they're associated with, or they're interacting with the water on both sides of the membranes, and very little or no water in the hydrophobic region. But here we have some proteins, the ribbon, using the ribbon model of the proteins. Again, we've still got phospholipids surrounding these, but the actual proteins that are inserted in the membrane are shown here in the ribbon form, and you'll notice that there are alpha helices here. And alpha helices are really important in proteins that are inserted into membranes, because if you look at a helix from the end, it looks like a circle. And if you divide that circle up into two hemispheres, and you say that one hemisphere is hydrophobic and the other hemisphere is hydrophilic, then you begin to understand how these alpha helices can be important in a protein that's inserted into the membrane. Because if it's inserted into the membrane, it's got to interact with the phospholipids somehow. And it's got to interact predominantly with the hydrophobic tails, because they form the inner part of the membrane. So there's got to be a hydrophobic region in the alpha helix. But what we also know is that a lot of these proteins form hollow channels, and we'll talk more about those in depth in one of the later sections of the presentation this morning. And that means there's going to be water present, which means they have to be hydrophilic as well. And so in many of these proteins that are inserted into a membrane, the alpha helix is divided into two hemispheres. One hemisphere is hydrophobic, one hemisphere is hydrophilic, to allow the inserted proteins to interact with both the phospholipids and specifically the fatty acids of the phospholipids, but also interact with water that is acting as a transport medium for things to pass through the hollow tube that's formed by these proteins. Now, that may sound fairly complicated, a fairly complicated description of what's going on here, but you'll learn more about this as we progress through the presentation. But this diagram comes very early in the textbook, and I just wanted to explain what this diagram is trying to illustrate. It looks like a little bit of a nightmare, someone's idea of a nightmare drawing, but in fact, it really does explain how some proteins insert into the membrane to carry out a particular function, something that we will talk about at length as we keep going. So I've talked about the phospholipids and the chemical nature of the phospholipids. So here is the bilayer that forms up spontaneously whenever we mix the phospholipids into water. And you can see how they arrange themselves. And this is showing it in a little bit more detail. And in this case, we're showing phospholipids that contain non-saturated fats or unsaturated fats. So these are the fats with the kings. And these do turn up in the phospholipids that are found in, but predominantly plant cell membranes, because remember, plant cells make a reasonable percentage of unsaturated fats. And so it stands to reason that those will also turn up in the phospholipids that form the cell membranes. But we also need to take into account the fact that animal cell membranes are made up of phospholipids that are going to contain predominantly saturated fats. And so there is going to be a structural and therefore functional difference between plant cell membranes and animal cell membranes. And yet they both have to have at the heart a basic function. So let's take a look at the phospholipid composition of animal cell membranes and plant cell membranes and see how we create a cell membrane in both types of cells that functionally have similar functions. So we will get, that composition diagram is coming up a little bit later, I believe. I'm getting a little bit ahead of myself. So there have been several models floated on what a cell membrane actually looks like in terms of a phospholipid bilayer with proteins. And the reason why there have been several models is because of the way initially scientists went about examining the chemical nature of a membrane. So once it was really recognized that phospholipids as lipids, as fats, were the main component of the cell membrane, the next thing was to try and isolate chemically the membrane components to see what they were like. And of course, we know that we can solubilize fats using detergents. And so that was the basic approach to solubilizing membranes. Use a detergent, and we actually have in research labs these days specialized detergents that are made specifically for doing this. Solubilizing detergents as a way of extracting out the phospholipids, extracting out the membrane so that you could take a look at them. The problem was that every time you did this, you always got proteins associated with it. And you could never quite separate the phospholipids from the proteins. And eventually it dawned on the researchers that were doing this, that the reason why these proteins were being solubilized with the fats, the phospholipids, was because they must have been a component of the membrane. That if you solubilize a membrane, not only do you get the phospholipids, but you also get the proteins that are associated with the membrane. So now it became important to try and identify where those proteins might be located. Were they an integral part of the membrane, or were they surface components of the membrane? And around the time that people were really starting to think about this, because they had done these chemical analyses, it was possible to start looking at membranes physically, because electron microscopes started to become common research tools. The first electron microscope started to appear around 1935 and started to become commonplace on university campuses and at research institutions in the early 1940s. And there are techniques that allow you to prepare the membrane so that you can visualize them in the electron microscope. And we still use those same basic techniques today. And what you see when you take a look at a membrane in the electron microscope, and I've done this many times, I believe I've told you that I used to make a living doing electron microscopy. And so I've seen my fair share of membranes in the electron microscope. And when you look at a membrane in an electron microscope, what you see is an empty space bounded by two dark staining regions on either side of this clear space. And these were interpreted as being the phospholipids and specifically the bilayer. And these regions, these dark staining regions on the outside, were interpreted as the protein layers. These were the proteins associated with the membrane. And out of this would grow what was referred to as the sandwich model of a membrane, where the phospholipids were sandwiched between two layers of proteins that were the proteins that were constantly being isolated every time you isolated the membrane. And it's also known as the Davson-Danieli model, after the two biologists who proposed this model. And it was built around the observation that you always isolated proteins with the phospholipids whenever you tried to solubilize a membrane. And when you got a look at a membrane in the electron microscope, this is basically what you got. And as far as the chemistry of the staining processes for electron microscopy, this seemed to make sense in terms of what you could see. It turned out to be totally wrong, however, because it was a misinterpretation or a misunderstanding of the nature of the staining. And a twofold set of data was again produced that changed everyone's idea of what the membrane looked like. And ironically, some of that data was again from the electron microscope, using a different technique that actually allowed you to look physically at the individual phospholipid bilayers, because it is possible to actually separate those using a technique which again I've played with. I'm not particularly good at it, but people who are really good at it can actually separate the two phospholipid layers of the phospholipid bilayer, and that allows you to take a closer look at the actual physical structure of the membrane, and you start to see that in fact the sandwich model is not correct. So what is the current model of the membrane? Well, it's called the Singer-Nicholson model, or the semi-fluid mosaic model. That's okay. So it's not accepting this. So let's just put in here. This is going to be a mess. Let me erase this and then let's start this again. So it's called the semi-fluid mosaic model, or the Singer-Nicholson model. And this is what we think a membrane now looks like. This is an artist's representation of what we think the model looks like, the membrane looks like. And you can see that rather than there being a layer of proteins on either side of the membrane forming this sandwich of phospholipids between the proteins, you see that most of the protein is actually inserted into the phospholipid bilayer. So why this change in the model? Well, the first piece of data that sort of, I'll put it this way, went against the sandwich model was the fact that the vast majority of proteins that were being extracted out of the membranes along with the phospholipids contained major hydrophobic regions. And if they have a hydrophobic region, the one place they're not going to be is on the surface of the membrane in contact with water. They want to be in a hydrophobic region. And as far as the membrane is concerned, there is only one hydrophobic region for a protein with hydrophobic areas in the protein. And that's the center of the membrane where you've got the fatty acids of the phospholipids. That's the hydrophobic region of the membrane. So any protein that's got hydrophobic regions of its own is not going to be on the surface of the membrane. It's got to be buried inside the membrane in association with the phospholipids or in association with the fatty acids of the phospholipids. And the vast majority of membrane proteins have these hydrophobic regions. So they can't be on the surface. They've got to be buried in the membrane itself. The second piece of data, and this is where the electron microscope came into play, was a technique called freeze fracture. Here you take cellular material and you instantaneously freeze it. And the best way of doing that is to use liquid nitrogen, which has a temperature of around -200 degrees Celsius. You flash freeze it. And what that does is stop the ice crystals that are going to form as the water freezes from getting very large, because everything freezes so quickly. And that stops the cells from bursting. It's the reason why frozen food, so if you buy frozen vegetables, when you thaw those out, they don't turn to mush because they were flash frozen using liquid nitrogen or carbon dioxide. Very, very cold material that instantaneously freezes everything and stops the ice crystals from growing into large crystals that would rupture the cells. But this is also the reason why when you freeze stuff in your freezer, when you thaw it out, it typically turns to mush because it slowly freezes. It doesn't flash freeze, it slowly freezes. The ice crystals get really big. And if that happens to be inside some fruits or vegetables or something like that, that you're freezing, it's going to rupture the cells because the ice crystals get so big that they rupture the cells. So what we're doing here is flash freezing things to keep the cellular material intact. And then we mount it in a special device that has a very sharp, very, very intensely cold steel knife. And we pass it through the frozen material. And when it comes in contact with the cell membranes fracture. It's kind of like shattering. So if you can see this on YouTube, you take a tennis ball, you drop it into liquid nitrogen, and when you drop it on the floor, it shatters into 1,000 pieces. Whereas if you bounce the ball before you put it in the liquid nitrogen, it'd be just a regular tennis ball. The difference is that you've frozen the rubber it now becomes fragile and it shatters in its weak points. And this is what happens when you pass this super cold, very sharp steel knife through the biological material, the cellular material. The cell membranes fracture or shatter at their weakest point, and their weakest point is where the hydrophobic fatty acids are in contact with each other, because the only thing that's holding them together is the fact that they're both hydrophobic. There's no chemical bond, it's just that they're there because they're both hydrophobic. And if you think back over all of the interactions that we talked about back in chapter 4 a little bit, and then in chapter 5, and a little bit in chapter 2 as well, when we talked about chemical bonding, the weakest interaction of all of these hydrophobic interactions, they're very, very weak. So the connections between the two layers of lipids in the bilayer is very, 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 very tenuous. So this steel knife can separate them very easily. And when you do that, what you notice is that on one side of the two layers, you're going to see bumps sticking out from the phospholipids. And those are the proteins that are embedded in the phospholipid bilayer. In the matching position in the other half of the bilayer, you're going to see pits, and that's where the proteins were pulled out of the phospholipid layer, the fatty acid layer. And so someone who is really, really good at doing this, and I've never quite been able to master it to this level, but I know people who can, they can actually peel open these membranes and put the two layers side by side. Now, you can't put these directly into the electron microscope, but you can create a metal replica using a device, and that metal replica replicates exactly the phospholipids, the inserted proteins, and the pits where the proteins were pulled out. And you can actually see these when you look at the two halves of the phospholipid bilayer laid out side by side like this. You can see where it's been unfolded, and you can see the proteins on one side have been pulled out. You can see the matching pits on the other side. And it really depends on which of the two lipid layers the proteins stick with as to where you see the bumps and where you see the pits. But you're always going to see matching pits and bumps, indicating that the proteins are embedded in the membrane. Then when you take a look at the two sides of, say, a plasma membrane, the side that faces outside, the extracellular side, and the side that faces inside, the side that faces the cytoplasm, you see this difference in the appearance of the two faces of the phospholipid bilayer that represents the membrane. We say that the membranes have sidedness. The side that presents to the outside world is different from the side that presents to the cytoplasm inside the cell, indicating again the presence of different proteins and the insertion and of different proteins into the membrane. And it is this evidence, more than anything else, that indicated quite clearly that the proteins are not physically sandwiching the phospholipids. No, they are integrated. They are inserted in amongst the phospholipids as an integrated constituent of the membrane. So this is a really good example of what we now consider to be the true model of a membrane. There are integrated proteins that are inserted into the phospholipid bilayer. It accounts for the hydrophobic regions found in these proteins. It also accounts for what we see in these freeze fracture replicas of the lipid bilayers, either the individual lipid layers or the different sides of the same membrane, the two sides of the same membrane. It's the only way we can account for this. So once we start thinking in terms of this phospholipid bilayer with the proteins inserted into the phospholipids, it now gives us another idea that we need to focus on, and that's the fact that this is semi-fluid. It is not a rigid structure. things are actually able to move within the membrane, hence the idea of a semi-fluid membrane. So once we start thinking about the phospholipid bilayer, we have to start thinking of, and the semi-fluid nature of the membrane, we have to start thinking about where that semi-fluid nature came from. And that immediately starts us thinking about the fatty acids that are the major components of the phospholipids. And the realization that the fatty acids themselves can contribute to the fluidity or lack of fluidity of the membrane. So when we recall that plants make unsaturated fats and make a substantial percentage of unsaturated fats, it could be up to 40% of their fats are unsaturated. We recognize then that the unsaturated fats have that kink because of the double bond, and that double bond is in the cis form within the fatty acid. And that means that these phospholipids are naturally going to occupy more space. They're going to be forced apart. That reduces the density of the fatty acids and makes something that is more fluid rather than being more solid and more viscous. And so the presence of these unsaturated fats in plant phospholipids naturally makes the membrane more fluid and less viscous. In animals, however, remember the fats are saturated fats and they pack tightly together, which means the membrane is going to be very viscous, not terribly fluid. And we need to change that because we know that animal membranes are just as fluid as plant cell membranes. And it's that fluidity that is key to understanding the properties of a cell membrane. So how can we go about maintaining fluidity in a membrane that's made up of saturated fats that want to pack really tightly together? Well, that's where cholesterol comes in. You should recall that cholesterol, when we talked about it back in chapter 5, was described as an integral component of animal cell membranes. And the reason why it's considered to be an integral component is because as an amphipathic lipid in its own right, having a hydrophilic region because of the carbonyl group located at one end of the cholesterol structure, and then the strictly hydrocarbon portion of the rest of the molecule, which is exceedingly hydrophobic, means that cholesterol can insert itself in amongst the fatty acids because it is hydrophobic, but still orientate itself to the hydrophilic, hydrophobic nature of the phospholipids that make up the phospholipid bilayer because of the presence of this hydrophilic, small hydrophilic region on the cholesterol molecule and this large hydrophobic region on the rest of the molecule. Cholesterol is in fact amphipathic like the phospholipids and then orientates itself in the phospholipid bilayer in the same way. But that of course then forces the phospholipids apart and means that the membrane starts to take on a more fluid nature, just as forcing the phospholipids apart using unsaturated fats does the same thing in plant cell membranes. And so we now have a reason why cholesterol is essential for healthy cell membranes in animals, because it maintains the semi-fluid nature of the membrane and means that the membrane can remain fluid even in lower temperatures, where you might expect the membrane to become more viscous, perhaps almost solid. Likewise with plant membranes. In plant membranes, where the plants are growing in cooler temperatures, there's going to be a high percentage of unsaturated fats in the phospholipids to maintain the fluidity. If, however, you take a look at plants that are growing in warmer temperatures, you might find a reduced percentage of unsaturated fats and an increased percentage of saturated fats simply because the membranes are going to be more fluid because of the warmer temperatures. This is the reason why tropical plant fats like palm oil, palm kernel oil, some types of coconut fats are solid at room temperatures because they are in fact saturated fats, because the plants can tolerate a higher percentage of saturated fats because of the warmer temperatures. The membrane remains fluid, and in fact, it's somewhat advantageous to have saturated fats in warmer temperatures. It's also the reason why we find unsaturated fats appearing in the fat composition of cold water fatty fish. They need those unsaturated fats, again, to maintain a semi-fluid nature of their membrane, and also to act as an antifreeze in their blood to stop their blood from freezing. So there is this temperature relatedness in terms of the types of saturated or the types of fats that are found, or fatty acids that are found in the phospholipids that make up the plasma membranes. We do see this correlation. Where the temperatures are cooler, you do see a higher percentage of unsaturated fats, particularly in plants, but also to some degree in animals. Where in animals, we maintain the fluidity in cooler temperatures more by using cholesterol rather than using unsaturated fats. But there is a case to be made for using unsaturated fats, particularly in cold water fish. Where the temperatures are warmer, saturated fats are not a disadvantage. And so you see saturated fats turning up again in animal cell membranes, not so much the unsaturated fats. We still have cholesterol, however, to maintain the fluidity. But we see the effect of warmer temperature, most particularly on plant membranes, where we see in warm temperature tropical plants a decrease in the amount of unsaturated fats percentage-wise, and an increase in saturated fats percentage-wise. And again, that's reflected in the nature of tropical plant fats. So this fluidity is obviously really important. We can demonstrate the fluidity of the membrane by looking at the movement of proteins in the membranes. And to do this, we need to first tag the proteins using colored molecules, colored dyes, that allow us to identify the position of the proteins. And while this looks like we are making something like a Frankenstein cell, that might have all sorts of interesting properties. In fact, this is a non-viable cell. We are merging together a human cell and a mouse cell, but this hybrid cell will not survive for very long. But it survives long enough for us to carry out the experiment and see evidence of the free movement of the proteins in the membrane. So you tag the mouse cells with one colored tag, or mouse cell proteins with one colored tag. You tag the human cell proteins with a different colored tag, and then you merge the cells together. And it's relatively easy to do this both chemically and electrically. You can get the lipid membranes to merge. If you allow these to sit for a short period of time, maybe an hour or so, they go from this initial hybrid cell where there is a clear delineation between the mouse end and the human end to a cell where there is an even distribution of the two different protein groups across the surface of the membrane, a process called diffusion. We'll learn a little bit more about this a little bit later in this chapter. But it is only through the free movement of these proteins in the membrane that this distribution, this even distribution, it's an equilibrium. This even distribution of the proteins across the surface of the cell could take place. If the proteins could not move, they would remain stuck in their original position, maybe move a tiny fraction, but still retain this uneven distribution of proteins. The fact that they move to an even or equal distribution across the surface of the hybrid cell tells us that the proteins have moved in the membrane. They're free to move. We even know the lipids themselves can move. They can move around in the membrane under very rare circumstances. The phospholipids can even flip from one side of the membrane to the other side of the membrane. Something that is not common, but has been known to happen. So there is a real solid case to be made for both the lipids and the proteins being free to move within the membrane, which supports the idea of the membrane being not solid, not viscous, but being fluid, allowing for mobility of things within the membrane. And as you'll see when we talk about cell respiration and photosynthesis, both of these processes are dependent on the ability of proteins to be able to move freely within the membrane itself in order for there to be connections between major protein complexes in membranes. It becomes a really, really important idea. So to sum up what we now know about the basic model of a membrane, we can take a look at the next diagram to see what the basic nature of a membrane, and particularly an animal cell membrane, will look like. So the basic foundation of the membrane is our phospholipid bilayer, with the hydrophobic fatty acids pointing inwards, the hydrophilic heads of the phospholipids pointing out. We have cholesterol inserted in the membrane to maintain its fluidity, so nothing that we haven't talked about before. Then we have two classes of proteins that are associated with the membrane. We have the integral proteins, the proteins that are inserted in the phospholipid bilayer. Again, nothing that we haven't talked about. The hydrophobic regions of those proteins are embedded in the hydrophobic region of the membrane. The hydrophilic portions of the proteins stick out and represent the bumps that we see on the surface of the membrane. These are sometimes referred to as transmembrane proteins because they span across the full width of the membrane. But there are also integrated proteins that emerge on one side but not on the other side, giving rise to the sidedness that we observe in many membranes. Then on the surface of the membranes, also contributing to the sidedness of the membrane, are these peripheral proteins. These are hydrophilic proteins. They want to be associated with water, so the only way that they can be associated with water but still be associated with the membrane is to be on the surface of the membrane. So these are referred to as peripheral proteins. And we're going to see the role of integrated and peripheral proteins, both fixed and mobile, in both cell respiration and photosynthesis. Then you're going to see a number of other proteins that are associated with membranes carrying out other structures, other functions. So some of these might be membranes, some of them might be recognition proteins, some of them may be functioning as receptors. Even some of the lipids are going to be modified so that they're used for recognition purposes. Anything that's involved in recognition probably has sugar residues and therefore becomes either a glycolipid, a lipid with sugars attached to it, or a glycoprotein, a protein with sugars attached to it. And then for an animal cell, there's also going to be the extracellular matrix, again, something that we talked about back in chapter 6. That is a loose arrangement of proteins and fibers of sugars and proteins complex together that serve to partially reinforce the cell membrane of a protein, but of a animal cell. But remember, these are not rigid as a plant cell wall would be considered to be rigid, made out of cellulose, a little wooden box. This is not a rigid structure, but it does serve to help reinforce the animal cell membrane. And there are connections between the cell membrane and the extracellular matrix, just as there are connections between the cytoskeleton and the membrane. So there is an integrated connection between the cytoskeleton inside the cell membrane itself, and then the extracellular matrix on the outside of the animal cell. It's quite a complex system once you start to recognize all of these connections that exist around both the cell membrane itself and the integrated components in the form of proteins that are part of the membrane. So there are a number of functions associated with proteins embedded in the membrane or on the surface of the membrane. This diagram gives you a list of some of those. It's also tabulated in the chapter, and you can take a look at that in the chapter or take a look at the slides that you can download from the chapter resources in Canvas in the week 4 module. If you take a look in the Chapter 7 resources, you can find the slides that I'm using here. You'll also find a link to the narrated presentation once I mount that. Of course, you're seeing that if you're seeing that right there, if you're watching it right now. So yeah, no point in talking about that at this point. So cell recognition is an important part of the sugar modified proteins and phospholipid and lipids. We're not going to talk substantially about the cell recognition at this point. We'll cover a couple of examples of cell recognition proteins and glycoproteins a little bit later in the semester, but this is basically all you need to know at this point, that there is a role to be played by glycolipids and glycoproteins in cell recognition. If you are going on to take our A&P courses, you're going to learn much more about recognition proteins. If you take our General Biology 2 course, we do talk about recognition proteins there because they come into play when we talk about embryological development as one of the chapters that we cover in General Biology 2. So at some point in the not too distant future, irrespective of which direction you happen to be going in, the Allied Health Sciences and the nursing program, or a bachelor's degree, micro degree, biology degree, something like that, you are going to learn more about these proteins in more detail in those courses as you progress academically. So let's talk about a brief example of receptor proteins and how important they are. By looking at this example of the receptor protein that's involved in allowing the HIV virus to enter what are referred to as CD4 cells. They are components of our immune system. And that's how the HIV virus actually escapes the immune system, by hiding out in cells that are actually supposed to be identifying and getting rid of it. If you wanted to design a strategy for escaping destruction by our immune system, this would be the strategy to come up with. And unfortunately, it's the strategy that HIV has developed as it adapted first to chimpanzees and then ultimately was able to adapt to our immune system when it crossed from chimpanzees into humans. There are, however, some fortunate individuals in human populations. It's around 1 in 100,000, I think, is the incidence. It may be even smaller than that, but the number that comes to mind from, and it's been a while since I've read the papers or watched the documentaries on this, but there are individuals who can be exposed to the HIV virus and never become, and are HIV positive because their immune system has started making antibodies against the HIV virus, but they never develop AIDS. And the reason why is because the HIV virus is incapable of entering the CD4 cells and destroying those eventually, and therefore destroying the immune system. And the reason why is because they have a mutation that knocks out part of the CD4 receptor complex. And this is called the delta mutation of the CCR5 co-receptor protein. So it's referred to as the CCR5 delta mutation. And it knocks out this CCR5 protein that forms part of the CD4 receptor complex. Now, for this mutation to be effective, it must have preceded the presence of the HIV virus in the population, because HIV has only been a significant issue for human populations since the late 1980s. Although looking at genetic data, we suspect that The time period when the HIV virus crossed into humans from chimpanzees was probably much earlier, probably as early as the 1910s, but possibly as late as the 1930s. But somewhere in that time frame is when HIV finally crossed into humans from chimpanzees in the form that would ultimately become the source of the HIV epidemic once it moved into cities. Where did that take place? Probably in West Africa and most likely in what is now called the Democratic Republic of the Congo. That being said, as a cause of an epidemic, it did not really start to take off as a cause of an epidemic until the late 1980s. And that's when it started to infect a lot of people. And out of that epidemic would emerge the presence of individuals that were HIV positive that never contracted AIDS. And once it was recognized that the reason why they never developed AIDS was because the virus never destroyed their immune system. And it was then recognized that this was because it never got into the CD4 cells. The question then became, why? Why do they have this mutation that prevents the HIV virus from getting into the CD4 cells? Where did this CCR5 delta mutation come from? And through some very clever microbiology detective work, and there are several documentaries that cover this process that you can find. PBS had a really excellent one as part of their Nova series. So you might be able to find it for streaming if you look. But through some very clever microbiology detective work, it was discovered that these individuals that have this mutation have one thing in common. They're all descended from individuals who survived the plague in Europe. Now, the plague was caused by, or is caused by, a bacterium called Yersinia pestis. And it first arrived in Europe in a big way around 1330 and arrived via Italy. And in the next 50 years, it went through the population of Europe and killed somewhere around 50 or 60 million people, around a third of the population of Europe at that time. It was a major, major health issue. And then it vanished for a while. It didn't go away. It just stopped. The pandemic stopped. And the bacterium would re-emerge periodically for about the next 300 years. The last recorded major outbreak of plague, Black Death as it was known, in Europe was in 1666 in London. And what stopped it was the Great Fire of London and the rebuilding of London consequently after the fire. But the plague would re-emerge periodically for about 300 years, from about 1330 onwards. But what was discovered was that over time, it became less and less virulent. Fewer and fewer people died of it. More people survived. Now, at the time, no one knew why. We do know why now. And the reason why is because of the CCR5 delta mutation. It turns out that Yersinia pestis affects the human immune system in exactly the same way as the HIV virus. It uses the CD4 receptor complex, and it binds to the CD4 cells and kills off those cells and collapses the immune system and makes humans vulnerable to the bacterium. And specifically, it is bubonic plague that is the most deadly. there are two forms, pneumonic plague and bubonic plague, and it's the bubonic plague that is the most deadly. And they used to say that you could be healthy in the morning and dead in the evening by dinner time because of the plague. It would collapse your immune system that quickly and kill you as a consequence of that. But because the plague was, every time the plague re-emerged, it was killing people who lacked the CCR5 mutation, it was actually favoring or selecting for individuals who had the CCR5 mutation, and they were surviving in the face of the plague, and their numbers increased over time. And that's the reason why the plague became less and less virulent, because more and more people were living because they had that mutation and therefore could survive, and their descendants had that mutation and could survive the plague when it re-emerged. Once it was finally recognized that good sanitation would prevent the re-emergence of the plague, it disappeared. It no longer became an issue. But people with the CCR5 mutation still existed. But their numbers in the population decreased because the plague was no longer selecting for them. But then along came HIV, and now we know that that mutation is advantageous in the face of HIV, because it's using the same receptor complex, and the CCR5 delta mutation changes the receptor so that the virus can no longer enter into the CD4 cells, and therefore it can't destroy the immune system for those individuals that have the mutation. We knew that it couldn't be a product of HIV because HIV hasn't been around long enough to select for individuals with the mutation at the frequency that we see the mutation. It had to be something that preceded the HIV pandemic. And the only thing that makes sense now from all the detective work is the plague, first appearing in Europe in around 1330, and selectively favoring individuals with the CCR5 delta mutation for the next 300 years or so, and building up the number of individuals with that mutation as a consequence of killing off individuals who didn't have the mutation. So you can see how a receptor protein, or the absence of a receptor protein in this case, might be a benefit. And in the case of our current health pandemic, the COVID virus, you may recall, or you may have heard, that the vaccines are actually using what is referred to as the spike protein. The spike protein is for HIV, and make sure the pen is selected, and I'm putting red circles around the spike proteins. It's the spike proteins that allow the HIV virus to bind to the receptor. And so if you can prevent the spike protein from binding to the receptor, in this case by modifying the receptor, or in the case of the COVID virus, making an antibody that binds to the spike protein that prevents it from binding to the receptor proteins that the virus needs to bind to infect us, then you can prevent the infection. And so that's why the COVID vaccines use the spike protein as the antigen, the protein against which you're going to make the antibodies. Because once those antibodies start binding to the spike protein can't bind to the virus receptor on the surface of the target cells, therefore there can be no infection. So again, by using our knowledge of receptor proteins, we can prevent infection. And so just as we have focused on the spike proteins of the COVID virus to come up with a vaccine against it, so people are now looking actively at the spike protein of HIV, and more particularly the CCR5 delta mutation, to see if there's a way to functionally take advantage of what we know about the CCR5 delta mutation to come up with a way of blocking the CD4 receptor from attaching to the HIV spike protein in some fashion. A lot of research is going into this, a lot of money, research time is going into this to try and come up with a way to potentially mimic the CCR5 delta mutation, or at least its effect, so that you come up with a way of preventing HIV infection. I'm not sure what the state of play is on that research, but I know it is a major research direction that is being followed to try and come up with a way of preventing HIV infection in the future. So obviously receptor proteins are really important. Then we talked about the fact that membranes do show sidedness. That sidedness begins with the transport of proteins, or the production of proteins that are going to wind up in the membrane in the endomembrane system, specifically the rough endoplasmic reticulum, and the way the proteins, whether they be hydrophobic membrane proteins or hydrophilic peripheral proteins, associate with the membranes of the transport vesicles, the Golgi apparatus, and ultimately associate with the plasma membrane, and therefore either get released from the cell or get inserted into the plasma membrane as a membrane protein. All of the sidedness can be explained by the progress of the membranes, of the proteins through the endomembrane system as components of the membranes of the transport vesicles and the Golgi apparatus, or as proteins that exist within the lumen of the endoplasmic reticulum, the transport vesicles, and the Golgi apparatus as they make their way through the endomembrane system. So we've covered quite a bit in terms of membrane composition. So let's just take a quick break. If you need to go grab a drink, grab a snack, bathroom break, just want to walk away for a little while before you come back and continue on with looking at membranes, now is a good time to do that. Remember, you can put me on pause, you can turn me off. I don't care, because I will still be here when you come back. I won't notice. So for those of you who are sticking around, here is the concept check question for this break. The temperature at which organisms grow can affect the lipid makeup of their cell membranes. How would the lipid composition of a native plant growing in a warm environment differ from the lipid composition of a native plant growing in a cool environment? You stop for a few seconds, maybe 30 seconds, while you give some thought to that question. So you could cue the Jeopardy music. Okay, so let's kill the earworm Jeopardy music and let's take a look at the question. So this is something that I actually discussed a little bit earlier when we were talking about lipid composition. Remember, it is the fluidity of the membrane that's important, and that fluidity is a function of the fatty acids that make up the phospholipids. And it is the presence or absence of saturated fats that make the difference. So if you've got saturated fats as the major component of the phospholipids, the membrane is going to be relatively viscous and more or less fluid. Whereas if you have a lot of saturated fats, unsaturated fats in the phospholipids, then the membrane is going to be more semi-fluid and less viscous. So in a warm environment, you would want something that is more viscous. You don't want something that is too fluid, otherwise the membrane is not going to maintain its properties. So you would expect that a plant growing in a warm environment would tend to have more saturated fats as components of the phospholipids. Whereas a plant growing in a cooler environment is going to need to maintain a more fluid membrane, and therefore is going to need a higher percentage of unsaturated fats to maintain that fluidity. So again, that's the reason why fats that are extracted from tropical plants and warmer environment plants tend to have saturated fats in their phospholipids, and their fats tend to be more solid. So if you are thinking in terms of plant fats that are naturally like that, you think in terms of palm oil, palm kernel oil, tropical oil is another name that's given to it, or some of the different types of coconut fats that people use. If it is solid at room temperature under normal conditions, then you can be very confident, even if it is a plant fat, that it has a high percentage of saturated fat in it. So it's going to have the same health properties as animal saturated fats. So that's just something to be aware of. So let's move on and start talking about what the consequences are of these particular structures that we've been associating with the membrane now will have for its function. So once we recognize that we have a phospholipid bilayer with an extremely hydrophobic center and a small hydrophilic layer on the surface, that's going to impose conditions on movement of molecules across the membrane. Specifically, molecules that tend to be hydrophobic and tend to be very small are going to find it very easy to pass through the large hydrophobic region in the center of the membrane. Molecules that are in any way hydrophilic or polar are simply not going to be able to make their way through that hydrophobic region. It's just impossible. So the rule of thumb for free movement across a typical cell membrane is that you must be small and you must be hydrophobic. So let me move the text box out of the way here. Let me just check the next slide. Yeah, so let me just put this in here. Move the text box across a little bit so I can write on the slide. And just let me change that. Let me just check and see what I've got. I think I've got it in red. Yep. So rule of thumb. Free movement across the membrane requires small size and non-polar. And we can put in here uncharged. Hydrophobic. All of these terms apply. So if you want to move freely across backwards and forwards across a membrane, then you must be small and you must be non-polar or uncharged or hydrophobic. Biologically, as you're about to see, there are only 3 molecules that can move across a phospholipid bilayer because of these rules. And those 3 molecules are carbon dioxide, oxygen, and surprisingly, water. And there are reasons why for each of these molecules. In the case of carbon dioxide and oxygen, it's because they really are small and they are uncharged. They are both non-polar. Yes, I realize that oxygen is extremely electronegative, but remember that when you have 2 oxygens sharing electrons, those bonds are non-polar covalent bonds. So the oxygen molecule itself is non-polar and is very small. In the case of carbon dioxide, again, you've got 2 oxygens involved, but they are on opposite sides of the carbon. They're at 180 degrees opposite to each other. And even though oxygen is substantially more electronegative than carbon dioxide, the fact that they're on opposite sides of each other means that the polar nature or potential polar nature of those two covalent bonds between the oxygen and the carbon actually cancel each other out. And so carbon dioxide is also a non-polar, very small molecule. And so it can move freely across a cell membrane. And that's in fact how oxygen moves into cells and carbon dioxide moves out of cells, by moving through the membrane by a process called simple diffusion. Now, the cell talks about passive diffusion. I don't like that terminology. It really shouldn't be called diffusion. It should be called passive transport. Because diffusion, by definition, is passive. So to say passive diffusion is not necessary. It's doubling down on the idea of passive. So it's better to say passive transport and talk about simple diffusion rather than passive diffusion. So at some point, I will edit the slides to make this into account, but I can make these changes here now. So it is better to talk about simple diffusion and passive transport, not passive diffusion, because diffusion is by itself passive. And the reason why is because of the definition. So I've got a definition sitting here, and it says that it is the tendency of molecules to spread out spontaneously from areas of low concentration to areas of high concentration. And the reason why this is passive is because there's no external, that's going to be horrible. Let's erase that. No external energy required. This is the reason why it's passive. Because if it required energy, it would no longer, an external energy source, it would no longer be passive, it would be active. So when we talk about diffusion as being a passive process, it's because it is the difference in concentration that is required. Now, I could show you mathematically that that also means that there is energy involved, and we will see later that diffusion does provide a source of energy for cells. And it's a very important source of energy, but the mathematical demonstration is not necessary. All you need to realize is that because you've got this difference in concentration, there is an energy source available in that difference in concentration. You don't need any external source of energy. The molecules will now move spontaneously from where they are to where they aren't, from an area of high concentration to an area of low concentration, and they'll do that spontaneously. So long as there is a difference in concentration across a cell membrane, oxygen can move into a cell and carbon dioxide can move out of a cell. And that's how these two gases move into and out of cells, because there is a difference in concentration. There is more oxygen outside than inside. There is more carbon dioxide inside than outside because of the role of cell respiration, consuming oxygen in the cell and producing carbon dioxide as a byproduct. And then the bloodstream carries oxygen to the cells and carries carbon dioxide away to the lungs for removal, keeping the concentrations high outside the cell for oxygen and keeping the concentrations for carbon dioxide low in the blood to favor the diffusion of the two gases into and out of the cells respectively. So why does water move across? This is largely a function of the nature of the inside of the cell versus the environment that the cell lives in. There is a lot of stuff dissolved in the cytoplasm, so it has a lot of stuff in there. And as you'll see a little bit later, having a lot of stuff dissolved in the water reduces the concentration of water. Whereas outside of the cell, it's mostly water with not a lot of stuff dissolved in it, which means that there are a lot more water molecules. Effectively, the concentration of water is high outside of the cell. And because water can move across the cell membrane spontaneously, there is a tendency for water to flow into cells. And cells need to control that. But equally well, water can flow out of cells again as well. Because remember, it's the difference in concentration that matters. Depending on where the difference in concentration lies determines which way the water is going to flow. In the case of carbon dioxide and oxygen, that's not so much of an issue, but the flow of water really is, as you'll see. We have to talk very specifically about the flow of water. So here we have a diagram that illustrates the movement of water across the membrane, the fact that if we have a high concentration of water on the outside and a low concentration on the inside, the water will start to move spontaneously across the membrane or the barrier here, so long as that barrier is permeable, which cell membranes are, until we reach the equilibrium condition. Now, you should recall that our definition of equilibrium is that the rate of movement in one direction is equal to the rate of movement in the other. And that is, in fact, what we see when we reach equilibrium, whether we're talking about movement of water or we're talking about movement of solutes in the water, things dissolved in the water. Once we reach equilibrium, we have a situation where the rate of movement in one direction equals the rate of movement in the other direction. But we can also think in terms of equal concentrations, but it would be inaccurate, or it would be a bad thing to start thinking in terms of equal concentrations. By virtue of the fact that as soon as you start doing that, you may lose sight of the fact that really what we're talking about is equal movement. So, oops, that's not going to work. Let's tidy that up again. So that's an important thing to remember. Yes, we can talk about equal concentrations here, and it's perfectly legitimate to do so, because the equilibrium condition with regard to diffusion does represent equal concentrations on each side of the barrier. But it is better to think in terms of equal rates so that you don't lose sight of what we're really referring to, because when we reach equilibrium, movement does not stop. There is constant movement, but it's the fact that the movement in one direction is equal to the movement in the opposite direction. That's the key here. Movement does not stop when we reach equilibrium. It's that the movement in one direction is equal to the movement in the opposite direction that is the key. When we think in terms of multiple molecules moving, each of them will move independently of the other to reach their own equilibrium. So let's add that in here too. Multiple. As you can see, I'm still struggling with using the pen. And again, very important observation, that molecules move independently of each other to reach their own equilibrium. There are some exam questions that I really like to ask about diffusion that involve you, or require you to understand this principle of molecules moving independently of each other to reach their own equilibriums, and what the consequences of that are in the devices. I may show you a couple of those questions. I know I've got a couple of those questions in the review sheets for this chapter. And it will cause you to think about what is actually going on, but that's a good thing, because that helps you understand the principle of diffusion. And I want to remind you again that diffusion and the nature of diffusion and how it works with cells is one of these ideas, like pH, that the nursing program and the medical school at UCF have identified as ideas that they really wish students would bring with them with clear understanding from this class and from the other classes that are prerequisites for those programs. Because if you don't understand diffusion, you are really going to struggle with A&P, you're really going to struggle in some of the nursing classes. It's an important concept that you really, really, really need to grasp absolutely and clearly. And that's the reason why we are spending some time today talking about it, because it's really important. We can't escape the requirement to know this. When we start thinking in terms of water, then we start thinking in terms of osmosis. Osmosis is the term that we give for the special case of water diffusion. And the reason why we have to think about water diffusion is because of this semi-permeable nature of cell membranes. In many cases, the cell membranes are not permeable to the solutes that are present in the solutions on the inside of the cell and on the outside of the cell. But water is always present, and cell membranes, remember, are always permeable to water. Water, remember, is one of the three molecules that can pass through freely a cell membrane. So even though the solutes may not be permeable to the cell membrane, the water always will be. And that poses a problem for all cells, because having solutes present in a solution changes the concentration of water. And there are three terms that we use to describe that. So I want to introduce you to these terms, even though we're not going to address those terms specifically right now, but I do want to introduce you to this terminology because it makes it a lot easier to talk about what's coming up. So let me move this text box a little bit higher up so that I can use this whole slide. So there is a term, and I'll change it to black, I think, tonicity. And this refers to solute concentration. So when we were talking about molar solutions a couple of chapters back, we were actually talking about tonicity of solutions because we were talking about concentrations. So when we talk about concentration, we're talking about tonicity. And there are three terms that relate to tonicity. One term is hypotonic. One is isotonic. And the other is hypertonic. So when we talk about hypo, and unfortunately all of these terms and their definitions come to us from chemists, and chemists are really into concentrations of solutes. We as biologists and as biologists thinking chemically, we are not so much interested in terms of solutes because, again, cells can limit the flow of solutes across the membranes. What they can't limit is the flow of water across the membrane. So what our interest is mostly in, is the concentration of water. But these terms all relate to concentration of solute. So they prove to be a little bit problematic in terms of understanding what they're all about. But so long as you keep in mind what these terms are all about and the relationship between solute and water concentration, we can use these terms and they're easy to understand. So hypotonic means low solute. Concentration. But it also means if there's low solute, therefore high water concentration. So if you're wondering why that's the case, let me just give you a mind example to help you understand this. So if you have a beaker full of just water, you have 100% water. The water concentration is 100%. As soon as you start adding some salt to it, say you add 5% salt. Well, now the salt concentration is 5% and the water concentration is 95%. So the water concentration has actually gone down as the salt concentration goes up. So the more salt you add, if you add 10% salt, well, the salt concentration is 10%, but the water concentration is now only 90%. It's going down as you increase the solute concentration or the salt concentration. So there is a correlation between solute concentration and water concentration. As solute concentration goes up, water concentration goes down. As water concentration goes up, solute concentration goes down, or vice versa. So that's how we can get away with using these terms, as long as we remember that. So isotonic means equal solute concentration. And so, as you might guess, also equal water concentration. So this would also be our equilibrium situation. But remember again, it's at equilibrium, not because of the equal concentrations, but because the movement in one direction is equal to the movement in the opposite direction. Movement doesn't stop, it's just that the movement is equal in both directions. Then of course, hypertonic, as you might guess, means high solute concentration. And therefore low water concentration. Now, where you run into trouble with these, and we can see this in the next slide, and this is where students first struggle a little bit with these terms, is that these terms are not absolutes. So if I give you a container of salty water and ask you which of these terms would you apply here, your first inclination is probably to say, well, it's hypertonic, it's got salt in it, so it must have a high solute concentration, it's hypertonic. But compared to what? There's the problem. These terms are not absolutes, these are comparative terms. You've got to know what you're comparing the solution, a solution against, to know whether it's hypertonic or hypotonic. So let's go to this next slide and I can illustrate that now that we've got the terms in place. So here is a tube that contains sugar solutions, two sugar solutions separated by a semi-permeable membrane. So it could be a cell membrane or an artificial membrane that behaves like a cell membrane. Over here we have low solute concentration, which means we have high water, and the term that we would want to perhaps apply here is hypotonic, but we can't say hypotonic because we only know what the sugar concentration is in this solution, but we don't know what we're comparing it to. But if we look at the solution on the other side, we have a lot of sugar. So we have a high solute concentration, low water concentration. So if we're comparing this solution with low sugar to this solution with high sugar, this solution now becomes hypotonic, that's ugly again. Sorry about this. Let's clean that out and make it neater. So this would be hypotonic compared to the solution on the right-hand side, but this solution would in turn be hypertonic. Compared to the solution on the left-hand side. Now, because this is hypotonic, it's also high water. And because this is hypertonic, it would be low water. So even though this membrane is selectively permeable and doesn't allow the sugar to move, it is permeable to water, and we now have a difference in water concentration. And you might recall that anything in square brackets means water, it means concentration, so that's water concentration from left to right. So water diffuses to the right to reach equilibrium. And because the salt can't move, the only way that it can reach equilibrium is for the water level on the right-hand side to increase. So that's the reason why the water is moving to the right. You've got a low water concentration, hypotonic solution. On the left-hand side, you've got a high solute high solute solution on the right-hand side, comparing each to the other. That's the only way this works, by making those comparisons. And that gives us a clue as to which way the water is going to diffuse. So here's another rule of thumb for you. Water always diffuses from hypotonic to hypertonic solutions. And because that's the case, you can always figure out which way the water is going to move, because it's very easy to figure out which is the hypotonic solution, which is hypertonic solution. A hypotonic solution, low solute; a hypertonic solution, high solute. Now you know which way the water is going to go, from hypo to hyper, and you should be able to solve any diffusion problem under those circumstances. It's not difficult under those circumstances. Then the other thing that you need to know is that in biology, and that would also include medicine, the cell is the reference for determining tonicity. So when we talk about, for instance, an isotonic saline solution, which you'll encounter if you're going into the health sciences in any way, whether it be allied health sciences or nursing, or you're going to medical school or something like that, even into vet school, you'll encounter isotonic saline. What does that mean? Well, it means that it is isotonic to our blood, and it maintains the consistency of our blood, and you'll see the reason why shortly. But isotonic saline is called isotonic saline because it is equal in salt concentration to the concentration of solutes in the blood. And it's very important that that be the case. Getting that wrong is going to create all sorts of issues. If you use a hypotonic solution or a hypertonic solution, you're going to create all sorts of issues that are potentially deadly. Understanding what isotonic means and making sure that whatever you place into the body is isotonic becomes of critical importance to maintaining the health of your patients, whether they be human or other animals. So this is something that will have extreme relevance for you in whatever health science you are or aspect of the health sciences you're planning to go into. This is the reason why the nursing program, the allied health sciences programs, the medical school over at UCF, and I'm pretty sure every other medical school in the United States wants their students to have a really solid knowledge of diffusion. So when we start thinking in terms of cells and their relationship to water, we have to think in terms of, well, does the cell have a cell wall or does it not? That's the big deal. If you have a cell wall, the cell wall can stop the cell from swelling and therefore stop the water from flowing in. So bacterial cells, plant cells, fungal cells that all have cell walls, they simply use the cell walls as part of their defense against water naturally wanting to flow into cells, which it will always want to do, because the environment of most cells is hypotonic compared to the inside of the cell. And so water will naturally want to flow in. If, however, the external environment is hypertonic, then the water will want to tend to flow out. But that's a different issue. For most cells, the issue is that water wants to flow in. So how to prevent that? Well, if you have a cell wall, you can do that. I'll show you a diagram that relates to that in a moment. But a lot of protists, and here you can see paramecium. So let me just put a label under here. This is paramecium. A lot of protists have these specialized vacuoles, and I talked about these in the last presentation when we talked about vacuoles. You can see here the contractile vacuole is in its contracted form right now. It has these branches that penetrate out into the cytoplasm. These are actively collecting water that's flowing into the cell and bringing it to the vacuole, which will start to swell as the water flows in. When it gets full, it then contracts down and pumps the water back out. So in the case of these protist cells, and particularly those that are referred to as ciliates, of which the paramecium is a great example, they have these contractile vacuoles. They typically have several of them. So there's another one over here that's not as clear. And these simply capture the water that flows in. The cells do not even prevent the water from flowing in. They simply capture it in the contractile vacuoles and pump it back out again. So this is a way of dealing with water flow if you don't have a cell wall. It's one way of dealing with it. Actively capture the water, pump it back out again. But that requires energy. If you have a cell wall, like a plant cell, like a fungal cell, like a bacterial cell, that cell wall prevents the cell from swelling. So given that water is going to have a natural tendency to flow in, that means the plant cell is going to swell up until the cell wall prevents that, which stops any further water from getting in. But the cell is now fully swollen, and we say that it is turgid. So this is the normal, and we need to make sure that we're aware of that. This is the normal plant condition. When a plant does not have sufficient water, then the cell becomes flaccid. And this is where the plant wilts. So you need to water the plant to get it to return to turgid again. This is not natural for plants. The cell dies if it gets plasmolyzed. That's as simple as that. So this is not natural for plants. We can get it to do this in the laboratory, but this is not a natural condition in plant cells. These are the two conditions that the plants tend to move backwards and forwards between. So if there's no water, it becomes flaccid. When you water, it becomes turgid again. And you can make the same argument for bacterial cells and fungal cells. As long as there's sufficient water, they remain turgid. For animal cells, the situation is a little bit different because they have no cell walls. So instead, they have to maintain an isotonic condition. And your body works really hard to maintain an isotonic condition. That's why isotonic saline or another solution called Ringer's lactate, which is also isotonic to blood plasma, is really important. Those are the solutions that are typically included in the drip bags that they put into the arm of every patient that goes into hospital to stop them from getting dehydrated or to rehydrate them if they are dehydrated, because they're isotonic and it maintains the normal condition of the red blood cells and the other cells in the bloodstream, but also the body cells, because blood plasma also becomes interstitial fluid, the fluid that leaks out of the bloodstream to surround all body cells. So it's extremely important. If you place a red blood cell into a hypotonic solution, it swells up and bursts instantaneously. It happens faster than you can check it under the microscope. It's an instantaneous swelling because the water flows in, it floods in. There are specialized proteins called aquaporins that allow the water to flow in to reach equilibrium. But of course, because the cell doesn't have a cell wall to prevent it from swelling up and bursting, it swells up and bursts. So having an isotonic environment is really important. The reverse, not having enough water or having too much solute, means that these swells shrivel up. And again, this is not normal. This is also lethal. So if your red blood cells swell and burst, or if they shrivel, both of those are potentially lethal conditions. This can happen if you drink too much water. It can lead to water poisoning. So water poisoning. And this is very real. If you drink too much water and your body doesn't pee it out fast enough, you can dilute out your blood to the point where it can become deadly. And it has become deadly in a number of situations that I have become aware of over time. The last time that I heard about this becoming deadly was a competition run by a radio station in Texas about 5 years ago. The prize was a new car, but the trick was that you had to drink as much water as you could without peeing. And this cost the life of the person who came second in the competition. Not only did she not win the car, but she died of water poisoning after she got home as a consequence of drinking too much water in a short period of time and not peeing it out fast enough. The reverse, the shriveling, would be if you were to drink salt water. That's going to suck the water in your digestive tract, is going to suck the water out of your body and turn your solid waste into liquid, and you get a bad case of diarrhea. My previous dog used to do this. When I took her to a dog park on Tampa Bay where they can run off leash on the beach, she used to see Tampa Bay as a giant water bowl. And she used to drink the seawater, and half an hour later, the consequences of that would happen all over the beach. And then I would always carry a couple of gallons of fresh water with me so that she could drink the fresh water to flush the salt water out of the body once she realized what she had done. She never learnt, because every time we went there, she would do the same trick over again. And the same thing can happen to us. And in some cases, it can be planned. So if you ever have to have a colonoscopy, which you actually will once you reach 50 years of age, it becomes a standard test that is done. One of the things that they do is the night before have you drink something that has a high solute concentration, and it draws fluid out of your body into your digestive tract, and it has the same effect as the salt water had on my dog, because I want to clean your digestive tract out. But if it's carried to the extreme, it can also affect your blood plasma temporarily. And if it's done in a controlled fashion under doctor's orders, it can clean out your digestive tract, so they can take a look at the health of your digestive tract. And then they encourage you to eat and drink. They don't actually encourage you to not drink, just as long as you drink clear fluids to keep your body fluid level up. They don't want solid food going into your digestive tract. They can deal with fluids in your digestive tract, not solid food. So they actually encourage you to drink clear fruit juices and broths and things like that, just no solid food when you get ready for the colonoscopy. So again, cells have different responses to hypo, iso, and hypertonic solutions, depending on whether they have a cell wall or not, whether they have a means for controlling the water flow. And the control of water flow is referred to as osmoregulation. So this slide is illustrating osmoregulation in animal cells and plant cells. How do you deal with solutions that have different solute concentrations? Well, if you're a plant cell, you use the cell wall to stop yourself from bursting. If you're an animal cell, you maintain an isotonic environment. If you are a protist, you capture the water and pump it back out again. All valid ways of osmoregulating. So that's the consequences of having a cell membrane made out of phospholipids. Some molecules can move backwards and forwards across the membrane freely if they're small and hydrophobic. All other molecules are prevented from getting across the membrane. Or are they? The short answer is no, they're not. The longer answer is it is selective as to which molecules can move backwards and forwards across the membrane. And what does the selection are the proteins that are integrated into the membrane. So the next type of transport is, again, a passive transport. And this is why I prefer the term passive transport rather than passive diffusion. I prefer simple diffusion, which we've just talked about, and then this type of diffusion, which is referred to as facilitated diffusion. Why is it called facilitated diffusion? Well, it's diffusion because a difference in concentration is necessary for the molecules to move across the membrane. But it's referred to as facilitated diffusion because proteins as facilitators of the movement are absolutely required. There's no external energy because you've got the difference in concentration. Remember, that difference in concentration represents a source of energy. And that becomes an important idea. But these transport proteins are essential because they allow the movement of the molecules across, or ions across the membrane. So the two types of facilitated diffusion exist because there are two types of transport proteins. One type is called a channel protein, which is essentially a hollow tube of protein formed by proteins that are inserted into the membrane. And this is where those alpha helices come into play that we saw in that diagram back at the beginning of the presentation. The other type of protein is called a carrier protein, and these proteins seem to defy the notion of passive transport because they change shape, and typically when a protein changes shape, we think of that as being work being done and therefore some form of energy required. But in this case, if there is energy being required to change the shape, the energy is from the diffusion gradient. So it's the movement of the molecules, the diffusion gradient, that actually provides whatever energy might be necessary to cause this change in the protein. So this is still movement along or movement down a concentration gradient. So this is really important. This is still diffusion. And the other key piece of this is that these proteins can move the solute in either direction. They move the solute in whichever direction the gradient exists. So perhaps I should put that in here too. Movement is in the direction. So it could be into the cell, it could be out of the cell. It really depends on which direction the concentration gradient exists. Is it a gradient into the cell, then the movement will be into the cell. Is the gradient out of the cell, then the movement will be out of the cell. These proteins don't differentiate between the direction of movement. All they look at is the direction of the gradient. That's the direction they move everything in. So that's really important to remember. Yeah, there's one more thing that I need to put in here. So let me move this. Movement of ions. Positive ions. So movement of positive ions is down an electrochemical gradient, which is movement following both an electrical difference and a concentration difference. And the electrical difference, it comes about because the inside of a cell is negatively charged overall. There are a lot of negatively charged ions in the cell, and as you'll see shortly, the cell also works really hard to pump positive charges out. As a consequence, the inside of the cell being negatively charged is really attractive to positive ions. And so positive ions move along this electrochemical gradient, attracted not only by the difference in concentration, but also by the negative charge on the inside of the cell. And so consequently, when positive ions move into the cell, their concentrations may actually increase to levels above equilibrium. But they're doing so because they're being attracted not only by the difference in concentration, but also by the difference in charge, the big negative charge sitting inside the cell. So that's an idea that we also need to bear in mind, because it becomes another important part of the movement of things across the membrane, particularly the diffusion part. And remember again, diffusion is one of those issues that everyone wants you to have a really good knowledge of when you move on to later classes. So that's another idea that you need to do a little bit more reading about in the textbook. There's a nice section in the textbook about electrochemical gradients as well. So time for another concept check, a quick one before we move on again. So why are carbon dioxide and oxygen able to cross cell membranes without the use of transport proteins. And if a cell uses a contractile proteins to move excess water, or if a cell that does that swims from an isotonic environment to a hypotonic environment, what happens to the activity of the contractile vacuole? Is it going to increase or decrease, and why? So, Jeopardy music. Okay, so in the interest of time, let's keep moving. We don't want to have this turn into a really long presentation, although it's going to be long enough. Why can carbon dioxide and oxygen cross the cell membrane? Well, remember that the rule of thumb is you have to be small, which they both are, and you have to be hydrophobic, which they both are. And there has to be a difference in concentration, which there is. There is a difference in concentration favoring oxygen moving into the cell and a difference in concentration favoring carbon dioxide moving out of the cell. So it's three parts. They're both small, they're both hydrophobic or uncharged, and there are differences in concentration that favor their movement in the directions that they move in. If a cell has contractile vacuoles and it moves from isotonic to hypotonic, what's going to happen? Well, in an isotonic environment, we wouldn't expect the contractile vacuoles to be very active because the water movement, the natural water movement, the osmosis movement of water would be equal in both directions. It's isotonic, equal concentrations. But if it goes into hypotonic, now the water concentration outside is higher than the water concentration inside, because hypotonic means low solute, but high water concentration. Likewise, the solution inside will be hypertonic, meaning high solute but low water. So the tendency is going to be for the water to flow in. That means the contractile vacuoles are going to have to become more active to take care of the extra water that's flowing in as a consequence of the hypotonic environment outside the cell. So if those are the solutions you came up with, good. You have a really good understanding of diffusion. If you struggle to come up with those, excuse me, answers, then maybe you need to go back and take another look at this section on diffusion and particularly simple diffusion, which would give you the answer for this. Anyway, moving on. We've talked about passive diffusion or passive transport, not passive diffusion, passive transport. Now we need to talk about active transport. So if we think in terms of what we've talked about as far as diffusion is concerned, diffusion has always been down the concentration gradient, and there's been no external source of energy involved. That's why it's referred to as passive transport. So active transport is transport that involves the reversal of both of those. So if diffusion or passive transport was down the gradient, active transport is now in the direction up the gradient from low concentration to high concentration. And there must be energy involved, primarily in the form of ATP. So those two things must be tied up in our understanding of active transport. There has to be movement against the gradient from low concentration to high concentration in this case. And there must be an involvement of energy from ATP as part of the process. So the classic example of active transport is the sodium-potassium pump, which is found universally in most cells, but very much so in nerve cells, because the movement of sodium and potassium into and out of nerve cells is an important part of understanding how nerve impulses are propagated down a nerve cell. You'll go into great detail on this in A&P classes and advanced biology classes, irrespective of which direction you're going in. So I'm not going to go into a whole lot of detail about nerve cells, but I do want you to understand the basic idea of what a sodium-potassium pump does, because it will help you come to grips with nerve cell function when you get there. And it's a really important part of your A&P class. You spend a lot of time talking about it. So the sodium-potassium pump, and let me move this box out of the way again. The sodium-potassium pump basically follows a cyclic process. So we could start just about anywhere in this cycle, but the obvious place to start is at the top of the cycle. And the reason why here is this is the logical place to start is because this is where you see the sodium going out. Now, cells do not like high concentrations of sodium. Sodium, it turns out, is fairly toxic to some of the important chemistry going on inside cells. So they like a little bit of sodium, but not too much. And so they're actively trying to reduce the amount of sodium inside the cell. The problem is that there are facilitator proteins that facilitate the movement of sodium back into the cells. So sodium is constantly diffusing into the cell, so it needs to have an active way of moving it out to keep the concentration low. By the same token, cells love potassium. Potassium is really important to cellular activity, so they like to maintain a high level of potassium. But again, if you have a high level of potassium inside the cells, facilitator proteins, particularly channel proteins, will naturally allow the high concentration of potassium to flow out. So cells want to constantly pump potassium in to keep its concentrations at relatively high levels. So it's trying to actively move the sodium out and move the potassium in. So it does so in parallel using the sodium-potassium pump. So when the pump is facing inwards, the 3 sodium binding sites are open, but the 2 potassium binding sites are closed. So the only thing that can bind to the pump are sodiums, which there are too many inside the cell anyway. So they quickly bind to the sodium sites. This triggers the transfer of a phosphate group from ATP, which, remember, is how you transfer energy. So the binding of the phosphate group from ATP transfers energy to the protein pump. It changes its orientation to face out of the cell. That automatically closes down the sodium sites, which forces the release of the sodium. There's only one place for them to go, out of the cell. At the same time, the potassium sites now open. So the only thing that can bind to the pump at this point are potassiums. 2 potassiums bind to the 2 potassium sites. That triggers the release of the phosphate. With the release of the phosphate, the energy is now removed. The pump reverts to facing inwards. That reopens the sodium sites, but closes down the potassium sites. There's only one place for them to go, into the cell. And so the cycle continues, as long as there is ATP present to power the oscillation of the pump to face out and then in and then out and then in to the cell. There's always going to be sodium ions to be pumped out. There's always going to be potassium ions to be pumped in. What is important to notice here, and this becomes an important part of recognizing what's going on here. Let's go back. The 2 ions being pumped independently hopefully you recognize what it is. It's independently against their gradients. So just as we see in diffusion that the molecules behave independently, so in this pump, the 2 ions are being moved independently of each other against their respective gradients. But there's a little bit more that we can glean out of this as we take a look at the way the pump is going. But we'll look at that in a little bit more detail in a moment. So just to remind ourselves of the different types of processes that we've encountered in terms of moving ions and molecules across the membrane. If they're small enough and hydrophobic, they can move by simple diffusion across the membrane, following a diffusion gradient, moving from high concentration to low concentration. If they're charged or they're a little bit too big to fit through the membrane, then they can move again by diffusion, but this time facilitated diffusion through transport proteins that facilitate the movement down the concentration gradient. If, however, the movement needs to be against the gradient from an area of low concentration to an area of high concentration, then that means you're moving uphill, you're moving up the gradient. That's work, you need energy. So anything that moves things against the gradient, active transport, requires not only a transport protein to facilitate the process, but also the use of energy to do the work of moving things against the gradient, to move things uphill. So hence the idea of active transport versus passive transport. So one of the things that the further ideas that we can glean out of the sodium-potassium pump. So in the sodium-potassium pump, 3 sodiums move out. And 2 potassiums move in. That is, more positive charges go out. And come in. This creates a more negative charge. Inside than outside. Such a difference is called a membrane potential. And is measured in millivolts. So it's an electrical charge. And the symbol for millivolts is lowercase m, uppercase v. It really is an electrical difference. And so the sodium-potassium pump is an example of what is referred to as an electrogenic pump, a pump that is creating this voltage difference, this difference in charge across the membrane. And this is referred to as the membrane potential. Now, the sodium-potassium pump is not the most important electrogenic pump of all. There is another one that's even more important, and that is called the hydrogen ion pump. That really does work very, very actively to move hydrogen ions from inside the cell to outside the cell. And again, it contributes to the electric charge across the membrane potential. But it's also creating a difference in hydrogen ion concentration. And again, we measure the difference in hydrogen ion concentration on the pH scale. And so there is actually a difference in pH across the membrane, the cell membrane, and it's measurable. It's around 1 pH unit, or a 10-fold difference in hydrogen ion concentration. The combination of this charge difference and the hydrogen ion concentration difference is referred to as the proton motive force, or PMF. So it's the combination of the membrane potential so that's the voltage or charge difference, plus the pH difference. Or hydrogen ion concentration. And the reason why we talk about this is because this is an extremely important energy source for cells. It's kind of like a cellular battery. You've got a negative end and you've got a positive end. And when you connect the negative and positive end of a battery together, you get energy out of it. If you want to take a small battery and take a piece of copper wire and connect the copper wire across the two ends of the battery, you'll find that that wire actually starts to get quite hot because of the energy that's being released as the electrons flow through the wire. Here, it's not electrons that are flowing, it's hydrogen ions that are flowing. So the proton pump and this movement of the hydrogen ions is so important, the cell actually commits energy in the form of ATP to actively pump the hydrogen ions out. To tap into this proton motive force, the PMF, we need to connect the positive and negative ends of the cellular battery together again. And the way you do that is to do it through a protein. The protein stands in for the copper wire connecting the two ends of the battery together. This allows the hydrogen ions to move back through the protein again as a quasi-diffusion process. It can sometimes be a facilitated diffusion process. Sometimes it's a little bit more active, but it is typically a facilitated diffusion process moving from high concentration to low concentration. But now we tap into the energy of that diffusing hydrogen ion gradient to provide energy to move other things like sucrose, which contains glucose and fructose, which under the right conditions in cell respiration could produce upwards of 70 ATP molecules. So you can see why committing some ATP to creating this hydrogen ion gradient would be a good thing for the cell, because it now allows to transport molecules into the cell that otherwise it couldn't, using the energy of the diffusing hydrogen ion gradient to increase the energy supply in the form of ATP. Commit some ATP to the process to get access to a whole lot more ATP from the sucrose, for instance. And the protein that does this is called a co-transporter. Now, why that's important is because in co-transport, the movement is, the two molecules is in the same direction. In a sodium-potassium pump, where the movement is in opposite directions, this would be an anti-transporter. And where the movement is in the same, is one molecule in one direction, this is called a uni-transporter. So there are actually three different types of transporter: a uni-transporter that takes one molecule in one direction, an anti-transporter that takes moves two different things in opposite directions to each other. And then we have a co-transporter that moves two things in the same direction with respect to each other. And that would be the movement of sucrose in parallel with the diffusion of the hydrogen ions. The hydrogen ions forming the energy transport or the energy source. And when we start thinking about cell respiration, and we start thinking about the source of energy for making ATP, this is it. It is the diffusion of hydrogen ions across a membrane that provides the direct energy source for making ATP. And the complex that does it actually looks like a proton pump running in reverse. So a proton pump is burning ATP to move hydrogen ions up the gradient. If we turn the proton pump around and allowed hydrogen ions to flow back through it, we could expect it to make ATP as the hydrogen ions move down the gradient. And we think there is some correlation evolutionarily to this idea of a proton pump and an ATP generating complex simply being derived from the same thing but being run in reverse. And there's even some biochemical evidence that suggests that. But we're not going to delve into that. I just want to point that out to you because there is a case to be made for that, and some people are actively investigating that. And there is some research that suggests that this is really the case. So, quick stop for another concept, break, referring to sodium-potassium pumps. So the sodium-potassium pumps of nerve cells create a membrane voltage across their cell membranes. It's really important for the propagation of nerve impulses. Do these pumps use or produce ATP? So I'll stop for about 30 seconds, cue the Jeopardy music. And of course, the answer here, it should be a no-brainer. These pumps, because they are sodium-potassium pumps and doing active transport, must use ATP, not produce ATP. That's it. That's the answer to this one. So we're getting to the final section. And it talks about, or it references, mass transport. So every transport that we've talked about up to this point has involved moving single ions or single molecules one at a time in a daisy chain, one after the other through a complex, or by passive diffusion across the membrane, or simple diffusion across the membrane. See, I'm even keep thinking about passive transport, and I shouldn't. It's passive diffusion or simple diffusion, and I prefer simple diffusion. So here we are now starting to think in terms of, well, what if we want to move our massive molecules across at once? Not individual molecules, but whole masses of molecules. How do we go about doing that? And again, it's the membrane that provides us with the solution to this. And so the two processes are called exocytosis and endocytosis. Exo means out. Endo means in. And then cytosis references the cytoplasm. Let's fix that. I was hoping I could just take out the S. Or cytosol. Remember, both words are interchangeable. So when we're talking about exocytosis, this is an important of large masses of molecules. When we talk endocytosis, this is the export of large masses of molecules. And you've actually already seen exocytosis, because the great example of exocytosis, and the one that everybody talks about in biology classes, is the movement of transport vesicles up to the plasma membrane to secrete proteins out of the cell that have passage through the endomembrane system. That's exocytosis, best example. In the case of endocytosis, there are three different forms of endocytosis. Phagocytosis, which translates as cell feeding. Pinocytosis, which translates as cell drinking. And then receptor-mediated endocytosis is a special case. Pinocytosis. So we'll talk about all of these three separately in a moment. So here are the diagrams that show you phagocytosis, pinocytosis, and receptor-mediated endocytosis. We'll talk about them separately as we move our way through. So here's phagocytosis, cell feeding. This is how phagocytes and amoebas capture cells in their environment. And you can see that it happens because the plasma membrane or the cell membrane invaginates inwards, and as it does so, it draws the particle, could be a cell, could be a clump of food molecules, into the invagination. And eventually it seals off around the outside, forming a food vacuole. A lysosome will fuse to release enzymes. And of course, those enzymes are going to be digestive enzymes to break down the food particle or whatever it is that's been encapsulated. Phagocytes, by the way, are members of the white blood cell family. They're part of our immune system. You've probably seen them at work, but you may not have recognized them because dead phagocytes around a cut form the pus that you see associated with a cut. Those cells are dying as a consequence of eating and destroying the bacteria that would otherwise be pathogenic to you. So they are literally sacrificing themselves to protect you from these pathogenic bacteria. But as a consequence of doing that, the pathogenic bacteria wind up killing them. So next time you see pus, now you know what's going on there. In the case of pinocytosis, the invagination and vesicle capture a random sample of the molecules in the cell's environment. So in this case, the cell is not being particularly picky about what it's trying to capture. It's just capturing whatever happens to be there in the environment. So it forms the invagination, which gets filled with the interstitial fluid, the fluid that surrounds the cell. And in turn, it includes whatever's dissolved in that fluid. That gets pinched off into the invagination that becomes the vesicle that contains the molecules that have been captured. If you want to get a particular group of molecules, then you use receptor-mediated endocytosis. So this is why it's referred to as a special version of pinocytosis, because again, it's an invagination of a coated pocket, but this time the inside of the pocket is lined with receptor proteins that capture the molecule of interest or as you'll see, a particle of interest, and allows that to be drawn into the cell. So you're basically enriching the cell in one particular molecule from its environment, or one particular type of particle from its environment. This becomes really important in understanding how we capture cholesterol particles from the bloodstream. So there's a condition, and it's a genetic condition, so it's inherited in families, called hypercholesterolemia. And it is often referred to as familial hypercholesterolemia. And that's where we get this notion that it is inherited, so familial. And it depends on whether you carry a mutation or not that affects the level of receptors that appear on the surface of your cells for capturing cholesterol particles. So cholesterol particles are present in the bloodstream or released from the liver, where they're made in the form of LDL, or low density lipoprotein particles. Low density. So that's what LDL stands for, low density lipoprotein. And there are a variety of particles. There are very low density lipoprotein particles, there are low density lipoprotein particles, and there are high density lipoprotein particles. The VLDLs and the LDLs are the problematic form of cholesterol, because if we don't remove those from our bloodstream, or we maintain high levels of those in our bloodstream, they can start to block the arteries, because they start to collect on the inner wall of the arteries, wherever the artery starts to get some inflammation and some irritation. And that will slowly lead to blockage of the arteries and reduction of blood flow. And if that happens to be in an important muscle, like your heart muscle, or in an important organ like your brain, you can have a heart attack or a stroke where tissue actually dies. That's what happens. It dies because it doesn't get enough oxygen. And those receptors are coded for by these genes. So if you have the normal gene for these receptors, you have a high level of receptors, and you sweep out the particles of cholesterol, and you maintain a low level of cholesterol in your bloodstream. Because we have 2 copies of every gene, and you'll learn more about this in the genetics section, of course, later in the semester. If one of them is the normal and one of them is the mutation for lower levels of receptors, you make a moderate amount of receptors, and you maintain moderate levels of cholesterol. And you can typically control that through diet and exercise and maybe medication. But if both copies of your genes for these receptors are for the mutant, meaning you don't have enough receptors or no receptors, then you have very high levels of cholesterol, and that requires substantial intervention, medication, and lots of blood tests over the course of your life, because you are at very high risk of heart disease and stroke, because you're maintaining very high levels of cholesterol in your bloodstream, which is very, very unhealthy. Likewise, it is unhealthy to have not enough cholesterol. So there is an upper and lower level of cholesterol, but most people have too much, not too little. And again, this is a mutation, and it does run in families, unfortunately. And it can cause all sorts of problems in families where the mutation is present. So this is another important part of understanding these mass transport processes. That, again, receptor-mediated endocytosis here is a really great example of where that becomes a health issue. And so is you, as a potential future health specialist in whatever level you are likely to have patients with these conditions. And it is important that you understand the significance of what you're dealing with. So one final concept check. because this is the last slide. We are at the end of this presentation. As cells grow, the plasma membranes expand to accommodate the increased volume. Which process, exocytosis or endocytosis, is responsible? And explain. Cue Jeopardy. Okay, seeing as how we are at the end and we're all here, presumably, let's just answer this question. So, which process, exocytosis or endocytosis, is responsible? Which is likely to remove lipids from the plasma membrane and which is likely to add lipids to the plasma membrane? That's all you really need to be aware of. And if you think about it, endocytosis is creating vacuoles that are moving into the cell. So it's going to be removing lipids from the plasma membrane, whereas exocytosis is transporting things from inside the cell to the plasma membrane. So the lipids of those vesicles, the transport vesicles, are going to be added to the plasma membrane. So when the cell grows, it's going to be exocytosis. So it's going to be the transport vesicles. And that's something that you would know if you remembered our talk about the endomembrane system in the last presentation. You would recognize that that would be the valid answer to this question. So we are done for this chapter, for this presentation. Again, if you have any comments, questions, or concerns about anything that I've covered in the presentation, by all means, come and see me in office hours or shoot me an email and I'll get back to you about anything that is concerning you. But if you think it's going to require a more extended conversation, by all means, come to office hours, because that's where we can do the best and most efficient work. Emailing backwards and forwards, playing email tag does not necessarily work effectively when we're trying to do a lot of explanation. So if you want a more detailed, more efficient way of doing it, by all means, come and see me during office hours. So until next time, when we will be talking about energy manipulation and enzymes as mediators of cellular chemistry. Bye for now. I'll see you in the next presentation.